Welcome back to Chapter 13, Bioenergetics and Biochemical Reaction Types. In this lecture, we will look at part 2 of this chapter. Here, we will continue our discussion on common biochemical reactions. Let us discuss the energetics of some chemical reactions. Hydrolysis reactions tend to be strongly favorable because they are spontaneous. Isomerization reactions, on the other hand, have smaller free energy changes. Isomerization between enantiomers usually happens at equilibrium. Complete oxidation of reduced compounds is strongly favorable. This is how chemotrophs obtain most of their energy. In biochemistry, the oxidation of reduced fuels with oxygen is stepwise and controlled. Most organic reactions fall within a few categories. First, cleavage and formation of carbon-carbon bonds. Cleavage and formation of polar bonds, which could be carbon, another heteroatom bond. Nucleophilic substitution mechanisms can happen, or addition-elimination mechanism can happen in cleavage of carbon heteroatom bond. It could be CO or CN or CS bonds. Hydrolysis and condensation reactions, internal rearrangements, elimination without cleavage, group transfers such as transfer of a proton, a methyl, or a phosphate. Oxidation reactions or oxidation reductions, and this is typical example of redox reactions wherein electron transfers happen. Let us first consider chemistry at carbon. The covalent bond between two carbon atoms can be broken in two ways. The first way is a homolytic cleavage, in which carbon-carbon bonds break into radicals, or carbon-hydrogen bonds break into carbon and hydrogen radicals. This kind of cleavage is very rare. The second way of cleavage is a heterolytic cleavage. It's pretty common, but the products are highly unstable, dictating the chemistry that occurs. In this case, a carbon-hydrogen bond can cleave into a carbanion and a proton, or a carbocation and a hydride. A carbon-carbon bond, on the other hand, can cleave into a carbanion or a carbocation, or a carbocation and a carbanion, depending on what groups that are attached to these carbon atoms. Biochemical reactions involve interactions between nucleophiles and electrophiles. Nucleophiles love nuclei. The nuclei of an atom consists of positively charged particles, or protons. So nucleophiles love to react with positive charges and are electron rich. Some examples of nucleophiles involved in biochemical reactions are O-, is negatively charged and is as in an unprotonated hydroxyl group or ionized carboxylic acid. Could be either of them. Negatively charged sulfhydryl group. Now if you remember enzymes, a serine hydroxy can be deprotonated to give a negatively charged O-. A cysteine thiol or sulfur can be deprotonated to give a sulfhydryl group. In addition to these two, there can be carbanions, uncharged amine groups, imidazole, for example in histidines, and simple hydroxide ions. Electrophiles, on the other hand, love electrons. Electrons are negatively charged, as you know. So electrophiles must be positively charged because they love electrons and are attracted towards electrons. Electrophiles, the common examples that are present in biochemical reactions are the carbonyl group. The carbon atom of a carbonyl group is electrophilic. Protonated imine group, this carbon is activated for nucleophilic attack. The phosphorus group of a phosphate is electrophilic, and a proton is electrophilic. Let us consider some common reactions that form and break carbon-carbon bonds in biological systems. The first example is aldol condensation. And in this reaction, a carbanion from one molecule attacks the carbonyl carbon of the next molecule to form an aldol product. The second reaction is Claisen ester condensation. Here, again, the carbanion attacks another electrophilic carbonyl center. And in this case, a thiol leaving group leaves. And this results in the formation of a carbon-carbon bond. For both the aldol condensation and the Claisen condensation, a carbanion serves as a nucleophile, and the carbon of a carbonyl group serves as an electrophile. The carbanion is stabilized in each case by another carbonyl. As you can see, this carbanion can resonance stabilize. It can go to this bond, and this bond can move up. In the third reaction, which is a decarboxylation of a beta-keto acid, what happens is that a carbanion is formed once this carboxyl functional group leaves. And that carbanion is again stabilized by the adjacent carbonyl group. And this is an example of a carbon-carbon breaking reaction. While substitution reactions from sp3 carbon proceeds via nucleophilic substitution mechanisms such as SN1 or SN2, substitution from the sp2 carbon proceeds via the nucleophilic addition-elimination mechanisms. Here, a nucleophile adds to the sp2 center, giving a tetrahedral intermediate. The leaving group eliminates from the tetrahedral intermediate. Leaving group may pick up a proton while leaving from the original molecule. Isomerization and elimination reactions happen in cells. And in those kinds of reactions, there is usually no change in oxidation state. For example, the conversion of glucose 6-phosphate to fructose 6-phosphate is catalyzed by this enzyme, phosphohexose isomerase. And in this reaction, as you can see, the starting material glucose 6-phosphate has a carbonyl on carbon 1 and a hydroxy on carbon 2. That just got interchanged in fructose 6-phosphate now, which has a hydroxy at carbon 1 and a carbonyl at carbon 2. If you consider the mechanism of this reaction, there are two residues. One is a base and one is an acid in the active site of phosphohexose isomerase. While the base abstracts a proton and polarizes the bond, a carbonyl bond, by pushing the proton inside and carbonyl abstracting a proton from the acid. This results in the formation of an enediol intermediate. And as you can see, enediol is 2-hydroxy that is attached to a CC double bond. Now, the acid that originally donated a proton to the carbonyl now acts as a base by abstracting the proton from the adjacent hydroxy. And that pushes the bond inside and results in proton abstraction of this enediol double bond from the original base, it was B1. This results in the formation of fructose 6-phosphate. As you can see, this is kind of an isomerization elimination reaction where there is no change in oxidation state.